so much for that kind introduction. Thanks for inviting me to talk. One of my favorite things is talking about, the fundamental biology of bacteria. It's something I've worked on during my PhD, and now that I continue to work on during my postdoc. And today, I'm going to tell you a really collaborative story, though. Something I've been working on the last few years with Susan in her lab in Norwich at the John Innes Center. But it's really a project that is really collaborative between our lab and the lab of Martin Pillhofer at the University of Zurich, or at ETH Zurich, excuse me, with this guy right here, Bastian Cassou. He's a really talented electron microscopist, and I'm going to tell a lot of the data that we generated together. But first, I want to, set your mind in the right space of what we think of when we think of bacteria. So I know this is a conference about bacteria orchestrating life, and I think that even when bacteriologists get together, we have a very, we immediately go to the sort of standard bacteria that everyone thinks of, which is E. coli. And so E. coli is, a pretty classic rod-shaped bacterium that divides by binary fission. And, it's sort of an almost original sin in bacteria when we talk about how bacteria work. A lot of the time people think of how E. coli work actually, and not actually how bacteria work. Because, right, even in bacteria, in the most basic sense, you have different shapes and sizes that you're familiar with. Maybe you've heard of Staphylococcus aureus or Treponema, which is, these sort of make the classic trio of bacterial morphologies. But bacteria really encompass a wide range of sort of shapes, lifestyles, and morphologies. And in this case, I'm showing you a couple of the Streptosporangium and Thermo bifida that make these large fruiting bodies, which produce cells of different types. Chetocytospora setae and Catenulospora acidophylla make spores. And then, of course, there's things like Caulobacter crescentus, which have, two different styles of living within one life cycle. But today I'm going to tell you about a bacterium that's near and dear to my heart, and that is that of Streptomyces silicolor. And so Streptomyces silicolor is basically my favorite bacterium. I've worked on it now for almost 11 or 12 years. And it's an interesting organism, but most people are familiar with it because of the antibiotics that it produces. And I believe Barry Wilkinson gave a talk yesterday about antibiotics, so I'm sure he mentioned the sort of awesome power that Streptomyces have to produce antibiotics. And in this case, I'm just showing you a few of the different colored antibiotics that we know that Streptomyces silicolor in particular, which is just one specific species of bacteria, that they produce. But Streptomyces are really interesting, not just because of the antibiotics, but because of their really unique and diverse life cycle. And so here's a graphical depiction of the life cycle of Streptomyces, where you start with a spore, right? A single spore, which is the resting cell of Streptomyces, right? It's just a single cell that is basically physiologically inert, that at a certain point during the life cycle, it interprets environmental signals and begins to germinate, which essentially means that the germ tube starts to form here. You can see it protrudes from this circular spore and starts to make a long filament. And then at a certain point, what happens is it begins to branch and divide. And you can see this in this electron micrograph on the side here. You can't make out the individual filaments because they're all wrapped together in this sort of mixture of extracytoplasmic matrix. And so, but they're making all of these filaments. And then at a certain point, what happens is the colony depletes nutrients and it interprets its own signals. And what happens is it pushes new filaments up from the main colony surface and forms these what we call aerial filaments or aerial hyphae. And these aerial hyphae are very different than vegetative hyphae because they actually have a very specific purpose, and that purpose is to be reproductive. And those reproductive filaments, what happens is you can see in this diagram right here, wherever you see this yellow line, that is where there's a physical barrier that comes up that essentially marks cell division sites. And so you can see in the next image, you can see there's a bunch of cell division sites that get marked all at once up and down the aerial filament. You can see that in this micrograph here where you have these filaments curling in on themselves. And what happens when they do that is they start to curl because they're about to divide into a chain of spores. And these spores can then go on and be spread typically by water, but you can also be transversed by air. And so you can see that these spores just become this nice chains on a bead, and that is sort of the start and the end of the life cycle of Streptomyces. But Streptomyces is unique for many reasons. So here's a graphical depiction of the Streptomyces chromosome. And the one thing you might think of when you think of bacteria is that bacteria have a circular chromosome, but you'll notice there's this big gap here. And that's because Streptomyces don't have a circular chromosome. They have a linear chromosome. And you can see these two little caps at the end. Those are terminal proteins that protect the ends of the chromosome. And so even at the genetic level, Streptomyces are quite interesting and unique. But that's not the only thing. And as I mentioned before, I believe Barry talked yesterday about the antibiotics of bacteria. And Streptomyces silicolor makes all of these compounds. These are not just antibiotics, but some are signaling molecules. And Streptomyces as a whole make antifungals, anti-cancer drugs. And the story that I want to tell you today from our work is actually a little bit different and unique. It's about how Streptomyces use these, what we call contractile injection systems, to modulate and respond to that developmental life cycle that I just told you about, and also to responses in the environment due to stress. And so you can see in this picture right here, it's maybe not very clear to you, but you can see that this is a model based on electron microscopy that shows each individual component of what's a very long internal molecule that I'll tell you a little bit more about. And so to familiarize yourself with contractile injection systems, what we have to think about is the setting the place. And so if you can see all of down here in this corner, it's not important that you know all of these things, but just to highlight what they are, you have this sheath, which is surrounding a tube, right? So this is a 3D, you want to think of it as sort of a spherical or a cylindrical object. This is a cross section of that. And so you can see on the outside, you have the sheath, and on the inside, you have the tube, and then you have this little spike at the top. And then usually there's some sort of membrane spanning compound, or membrane spanning complex, excuse me, that helps this tube cross the border of the membrane. And so what happens is that it starts with the type 6 secretion system, and then there are other types of systems that are not as embedded in the membrane. They can be brought to the membrane. And then there are molecules that exist outside the cell, which we call extracytoplasmic cis, or E-cis. And the whole purpose of these things are to basically compete with help bacteria compete against other bacteria. So if you notice here, you see they have a spike. And this is exactly as intuitive as you would expect it to be, which is this spike is meant to go through the host cell and into a different cell. So this is a depiction of a different cell over here. And so what that does is when it pierces that into that other cell, it releases a payload of toxic molecules and proteins to help destroy the enemy cell. And so the purpose of this, which is very interesting, is that you can see, if you're familiar with your basic bacteriology, this is a gram-negative cell because it has an inner and outer membrane and a thin layer of peptidoglycan. And for the last 20 years or so, or maybe even more, people have studied the type 6 secretion system, and it's been almost exclusively in gram-negative bacteria. And only in recent years have people started to look at the E-cis, which are the extracytoplasmic ones. And in the last couple of years, people identified, this is just a tree of life, that's looking exactly for those E-cyst loci that I just told you about in bacteria and archaea, the majority of them are still found in gram-negative bacteria. But if you notice this big orange brick here, that big orange brick is actinobacteria. And the vast majority of the bacteria in this block are streptomycetes. And so there are other gram-positive bacteria on here, like the firmicutes, but the majority of the ones that we see in gram-positive bacteria are streptomycetes. So the question is, if streptomyces express these, and when do they when and where do they express these? And so if we take a look at the streptomyces chromosome, in particular the streptomyces silicolor chromosome, we can look for this gene cluster. And it's not important that you know the numbers or the letters, but if I show you this graphical depiction, you can see that the genes that we would expect to encode the proteins of the cis cluster are in these color coordinated. So you can see the orange one and the green one are the sheath and the tube. And so you can see from this this gene cluster, you can see we can make up most of the cis opera or the cis structure that we would expect to have. And you can also see that it has enough proteins to actually complete the task. So it should be able to make not just a cis, but a functional cis as well. And so one of the things that we use, the techniques that we use, and particularly Bastian uses, is cryo-electron microscopy, which uses an electron microscope to basically blast our samples of streptomycetes or extracts of streptomycetes with electrons to see and look for these massive structures inside and outside and around the cell. And to do that, basically, it essentially uses a beam of electrons. And when you hit something that's very dense, it becomes dark on a micrograph. And if it's not dense, then it looks very light, which the surrounding cells will do. And Bastian actually uses a very specific type of electron microscopy called tomography. And what tomography does is it basically lets you turn a sample on its axis about 60 degrees in either direction, and you hit it with electrons the whole time, and you take images at each tilt. And what we can do using computer technology is recreate a three-dimensional image from all of these individual 2D images and essentially create a tiny little movie. And this will be important as I go on during the talk. And so to start with, what we do is we look to see, do streptomyces even produce these? And so just because the streptomyces has the genes to do it doesn't mean it always makes it, particularly in the lab. And so what we did is we created a mutant strain of streptomyces that removes that whole operon that I showed you on the other side. And when we do that, when we use electron microscopy, you can see these little bricks. It's not very impressive right now, but I promise it'll look a little bit better in the next one. And we believe these bricks are those contractile injection system. And if you notice in the cis mutant that where we deleted that gene, you don't see that. And we can look at this another way using a Western blot. And so a Western blot is used to detect specific proteins. And we made two different antibodies against very specific components of the E-cyst cluster that we thought. And you can see that both of them are in the wild type, which we would really expect. And so this is just taking the whole cell extract. If we grow a big culture of streptomyces and we just burst them open, then we can see these two proteins. And when you delete the cis, you don't see those proteins anymore, which is very good. And to do a really proper bacteriological control, you add the cis cluster back into a different part of the chromosome. So you do what's called complementation. And when we add it back, you see those proteins again. So it suggests that these proteins that we don't see, this complex that we don't see here, is actually the cis cluster that we wanted. But the thing we noticed that was very important is that I initially told you that we saw these based on the E-cis cluster. So they should be outside the cell. But what happens is when we look at the supernatants of streptomyces cultures, so when you just take the liquid that they're growing in, you never see these E-cis particles. which led us to believe that these are actually made and used entirely within the cell. And let me show you a tomogram. So this is that tomography that I told you about before. I don't know how well you can see this, but this is a slice of a cell. So here is the cell wall on the outside. And then additionally, you have the membrane on the inside, and it makes this sort of weird invagination. And if you look here, you can see these two really long bricks. And those two really long bricks are turned on their side in this image right here. And basically, using electron microscopy, we can actually make out some of the shapes of what structures we believe them to be. And so we can actually see these long, uncontracted structures totally floating just casually within the cytoplasm. And using subtomogram averaging, you can make these fancy models, and we can actually see and map each individual protein into this. And there are 60 layers of sheath and tube proteins here that show us we can see each individual component. And so the question then is, when is the cis cluster expressed? Because I told you that Streptomyces has this life cycle, and it's generally broken up into two phases. One we call vegetative growth, and one we call reproductive or aerial growth that ultimately leads to sporulation. And so on agar plates, Streptomyces celicolor takes about 4 days to go from a single spore to chains of spores at the end. And so if you look at a Western blot, again, where we're looking at the total cell content of our two proteins that we care about with the cis, you can see that in the first 48 hours, you see a ton of protein being made of the cis. And that 48 hours corresponds to vegetative growth. You do see some lingering about into 72 and 96 hours, but that's basically because of how Streptomyces grow. And we can talk about more about that if you have questions afterward. But so we are fairly confident, due to this and some data from a different lab that works on this as well, that these were predominantly made during vegetative growth. But the big question you might be wondering is, what actually do these things do in Streptomyces? So I'm going to tell you just highlight a few experiments because we don't have time to talk about all of it. But what I can tell you is that in Streptomyces, a really quick and easy way to look at Streptomyces is if you take a glass cover slip and you just place it on top of an agar plate, those spores of Streptomyces are so hydrophobic that you just put it on and you lift it up and it takes a ton of spores away with it. And as you might remember from the previous diagram, at 48 hours, you wouldn't expect there to be any spores, right? Because there was not making any of those aerial hyphae. At 72 hours, you do get a few here and there, but it's not until about 96 hours, about 4 days, that you start to see a ton of spores. And these are just standard light microscopy images. But what happens is when you delete the cis, or you make a version of the cis that's completely non-functional but still present, you see there's a ton of spores being made already. And when we complement that, right, by adding that cis cluster back at a different location, you can see that it goes back to the wild type phenotype, which suggests that this mutation that we caused is responsible for that phenotype. And we can quantify this by essentially counting colonies. And so if I draw your attention to just this main time point, then you can see that this is a logarithmic scale, right? So these are many orders of magnitude more spores found in the cis mutant, which suggests that the cis mutant actually causes Streptomyces to sporulate faster, which means that when you have the cis, the contractile injection systems, it delays the developmental life cycle. And if we go back to our electron tomography, again, I can't tell exactly how well this comes out, but we have a very healthy cell here with peptidoglycan on the side and a nice membrane that's in red. So if you look at this graphical depiction here of the tomogram, you might be better off to see these things. And so if you look at this, you can see these green structures. Those are the uncontracted cis. And in healthy cells, you only see those. If you start to see these membrane-bound cis, they are basically these cells that have membranes that are disrupted. You have a mixture of cysts that are in blue. Those are the contracted ones. So that means they've done their job, and the green ones. And if you look at a ghost cell, they have no membranes and all of them disappear and they become basically all contracted. So the question is, if you have only intact cysts in intact hyphae and you have only contracted cysts in dead hyphae, do they cause this, or are they like this because of that? So we tested membrane stress by using a confocal basically approach. And what happens is that we express a strain of Streptomyces that has GFP in the cytoplasm, which is very green. And all healthy cells will have green signal, but all cells will have a red signal because we stain the membranes with a dye. And what happens is that if you do a ratio of those, you should expect in a healthy culture, almost all the cells will be alive. So the ratio should be around 1. And so in the wild type, you have just this signal around 1, and in the cis mutants you have the same thing, so there's no difference. But if we use a drug, a sub-inhibitory concentration of the drug nisin, then what happens is you have the wild type, which has this severe loss of life. You can see it's dropped almost 50 or down to about half. Almost half the cells are dead. But the mutants of the cysts do not exhibit this, suggesting that the cysts are actually responding to the stress on the membrane and killing off the cells. And the take-home message here, just to wrap up very quickly, are that the cysts are involved in Streptomyces development, but also in response to the membrane-related stress that they are caused from the external environment. And if I have time, I'm just going to show you a very brief video depicting what we think looks like happening. And so if we go inside the hyphae here, we have cyst particles floating in the cytoplasm, which I showed you. And then what happens is there's what we believe a membrane adapter, which I didn't have time to tell you about, that helps it come to the membrane. And it interprets this stress signal and brings the cyst to the membrane. And what happens is those cysts then push through the membrane and eject their payload, which ultimately helps to cause lysis and ultimately cell death of those hyphae. And so with that, I'd really like to thank my lab, Susan in particular, who worked on this project with me, and also the lab of Martin Pilhofer, particularly Bastian, who has done basically all of the electron tomography that I got to show you today. And basically, we've worked on this collaboratively for the last like 3 or 4 years. And they have honestly been some of our best and most consistent collaborators. And I'd really like to thank you all for your attention, and I'd be happy to take any questions.
Thank you. Thank you very much, Joe. I would call you Joe.
That's perfect. Yeah, I go by Joe generally.
Thank you very much, Joe. Actually, before giving the chance to the audience to ask question, just a curiosity. So when you said mutant, so you said CIS mutant grow faster or less? So then
So
what?
yeah, so the mutants of the cyst. So when we delete the genes that have the cyst, that's the mutant that I'm referring to. They tend to go through the life cycle faster. So when you have the cyst, it seems to delay development. So the normal job of them seems to be prolonging the life cycle.
Okay, so mutant, but you mean by mutant because to avoid misunderstanding, because I'm a cancer researcher and sometimes
Okay.
some notion that so mutant, you mean by mutant, they do not have the CIS.
Yes. Yeah. Okay. Yep.
Okay. Question from the audience, because I have some, and I will tell Joe them later. So do you have question here from the audience? Yes, please. Ija, come. Wait, up.
Good morning, sir. So I want to first thank you for your presentation. It was very informative. But I also have a question. So for these cysts are like nano syringes, right?
Mm-hmm.
Okay, so are we able to exploit this technique to create nanotherapy, for example, for the
So that's a great question. What was your name? Sorry.
yes, ma'am.
Yasmin. Oh, thank you for that question. And so the sad part about that is you always hope that it's going to be, and currently as they are situated, the answer is no. At least not in the sort of sense that you might think. In that basically the problem we have is these contractile injection systems are very sensitive, and by that I mean that they are very trigger happy. And so when you extract them from streptomyces or really any organism, they almost immediately fire. And so it's really hard to get an uncontracted cyst. And so it's really hard to deliver them as a payload, sort of like as a therapy. That doesn't mean people haven't tried and aren't trying to sort of do that in some ways, and perhaps in more of a competitive bacterial sense. But yeah, as they're situated, no, you really can't extract them and sort of like use them to deliver something, unfortunately.
Maybe we can use them as a model to create, yes.
Yeah, that would be that's always possible. Yes.
Thank you very much.
Thank you, Yasmin. Have a nice day.
Any other question from the audience? Okay, so again with this CIS, so I saw kind of picture with the electron microscopy. So when cells are dead, so
Yeah.
this tube are not degraded; they are just contracted.
Yep.
So is there any? I mean, I'm just curious why they are not just degraded, not.
So that's a great question. So we know that in type 6 secretion, that very thing first thing I showed you, that they are regularly and rapidly recycled, and so they generally are degraded. With the ESIS, it's a bit more complicated because we don't think that mechanism necessarily is evolved to exist, because normally they would be released into the wild. And so you don't care at that point, right? You're not going to get those resources back. And so since we think this is from that lineage, it seems that it's like, okay, well, they're in the cell, but that maybe wasn't what they were supposed to be doing. And so there's no mechanism maybe to recycle them. It's possible there is, and we just don't see that. And it's just that some of these, the ones that we do see, are just the ones that are left. And so it's possible that some of them are recycled, but yeah, it does seem like they just kind of linger about after death.
True. Well, so last question, maybe, because we are after the morning conferences, we are doing also workshop in plasmid DNA.
Yeah.
I'm just also curious to know if the gene, I do assume it's a genomic gene, and do you think that we can, in a way, put it in a plasmid and put the plasmid in another type of bacteria to see a similar phenotype? I don't know.
So it's funny. So I actually I was a little light on the details, and I could have talked a bit more about the plasmids that we use. So I've made many plasmids for this this work, and we've expressed various components of the cyst within E. coli, for instance.
Yeah.
And one of the interesting things is, if you remember from the video, let me see if I have the slide here real quick. Oh yes, so the membrane adapter protein, I don't know if you can see this, is sort of what we think of is found embedded in the membrane. And what we did is we actually, if I can find it, yes. So I expressed this gene in E. coli just to see where it was. And so where it was in whether it was inside the cell or was it hanging outside the cell. And what you can see actually in this diagram is that using a plasmid to express this protein, you can see that the wild type E. coli, which is in this deep blue color here, there's the gene that we care about is expressed. It makes a much lighter blue color. This is, of course, fake staining, but it actually is representative of the fact that it doesn't really like this gene. And so it's growing quite slowly and quite poorly. And for some reason, even though it's embedded in the membrane and it's sitting on the outside, it is actually affecting the cell. And this is just the membrane adapter. So this isn't even the whole cis structure. Now, expressing the whole cis structure would probably be much more complicated because it's a very large operon, and it's actually multiple operons. So you probably wouldn't be able to necessarily assemble it in E. coli. I mean, you could try. But it's would be sort of complicated because you'd have to change all the promoters essentially, but you could potentially do it. But yeah.
Joe, thank you very much. It was very informative, very wonderful science, very informative. Thank you very much.
I appreciate that. Thank you
very much, and we hope to see you here in Tunisia for in situ presence and a wonderful talk. So thank you very much,
Wow.
and hope to see you soon.
Well.